Hello to everyone, I'm Paulo Ghia from the University Vita Salute San Raffaele in Milan, Italy, and I'm here at SOHO 2016 annual meeting presenting on MRD in CLL clinical implications. This is a very hot topic in the field, just because on July 1st, 2016, the EMA, the European Medicine Agency, has recently amended the guideline on the evaluation of anti-cancer medical products, and in particular they revised the guidelines for minimal residual disease detection in CLL. And they approved the concept that MRD detection in CLL can be used as a surrogate endpoint in clinical trials to achieve early and faster approval of new drugs. Why is that happening? Because as we know, since many years, at least more than a decade, we improved a lot the type of responses that we are able to obtain in our patients, thanks to combination regimens like FCR, where we are able to achieve a complete remission in the vast majority of patients, and also we are able indeed to achieve MRD negativity. So first of all, let me remind you what MRD negativity means. It does not mean cure, it does not mean absence of disease. It is simply a technical limit. It means that by the techniques that we are currently using, we are not able to reliably detect less than one cell in 10,000. So this is the limit, that is the current limit, and it is something that has been also reported in 2008 in the IWCLL guidelines. So one cell out of 10,000, which means 10 to the minus 4, and this can be achieved by using either 4-color flow cytometry or allele-specific PCR. Why has MRD been approved by EMA as a clinical endpoint for clinical trials? Because indeed in the past a number of regimens, a number of treatments in CLL show that patients who achieve MRD negativity, they score better in terms of progression-free survival and overall survival as compared to those who do not achieve MRD negativity. And this is true even with single agents like alemtuzumab, or with more complex regimens like FCR or FC, as you can see in the bottom row. The interesting point from the bottom row figures is that MRD negativity is associated with a better progression-free survival and a better overall survival, regardless the type of therapy that you use to achieve MRD negativity. So both patients treated with FCR and FC, if they achieve MRD negativity at the level of 10 to minus 4, then they have a better progression-free survival and overall survival. Though the MRD can be used as a surrogate endpoint in clinical trials, this should not be used in clinical practice, and this is also strongly highlighted and underscored by the EMA in their documents. And indeed, this is because though MRD negativity is a predictor of progression-free survival and overall survival at cohort level, at population level, we cannot use it at a single level of our patient. And indeed, if you see, for example, this study from the MD Anderson, patients who achieve MRD negativity and do carry also mutated immunoglobulin genes, they do have a very nice progression-free survival and overall survival, while instead, though they achieve MRD negativity but they carry unmutated immunoglobulin genes, they tend to have a much faster progression. So it's really suggesting that MRD negativity cannot be used as a single surrogate in every single patient. As I said, MRD is now acknowledged as a surrogate endpoint for clinical trials, but the dream of us as physicians would be to use MRD to guide the treatment in our patients, and maybe to aim for MRD negativity in our patients in order to go more closely to a cure. But this is not yet demonstrated, and indeed at this point we cannot say that if we do not achieve MRD negativity we are harming our patient. It is true that patients achieving MRD negativity, as in this study, CLL 11, patients treated with chlorambucil plus GA101, if they achieve MRD negativity, the top line, they score better in terms of progression-free survival as compared to those treated with rituximab plus chlorambucil. But patients who are treated with ibrutinib, they do not achieve MRD negativity, but still they have a very good progression-free survival. So really suggesting that MRD negativity should not be the aim to reach in every single patient that we are treating. So anyhow, in clinical trials, the use of MRD to guide treatment is explored. Here I show you one representative study, the CLL-M1 study, which is focusing on the patients that have a poor prognosis after FC or FCR, so the patients who are not achieving MRD negativity. who are not achieving the level of 10 to the minus 4, and these patients are randomized to either a placebo or a maintenance treatment with lenalidomide in order to improve the quality of response and the depth of the response itself. Another way of thinking about MRD-guided treatment would be also to assess MRD in order to decrease the quantity of therapy that we are applying to our patient if they achieve earlier MRD negativity. Having said that, you can find more on my full presentation, but I just want to remind you that the future of MRD in CLL is actually a present now, so we can use it, but please remember that it can be used only in randomized clinical trials. Do not use to treat your patient and to follow up your patient. Thank you very much for your attention.